Okay, we're recording. And today we're going to start talking about the nervous system. So, oh, that's a mess. Like all our systems, it's a really handy thing to remember if you can just kind of summarize the responsibility of a system in one or two sentences, just to sort of get you started. So when we think about the nervous system, it's one of our regulatory systems. It's one of the systems we have available to us to essentially regulate and monitor and correct homeostasis. So we know that homeostasis is maintaining biological values within upper and lower limits, and that the mechanism that's used is negative feedback. So this nervous system is one of the systems that is typically always monitoring conditions inside the body and outside the body and making adjustments in order to maintain homeostasis. And the other regulatory system is the one that we're going to talk about after we get the nervous system done, which is the endocrine system. These two systems both use chemical messengers. They both use messengers, otherwise known as signaling molecules, that you have chosen, most of you at least, have chosen for a topic by now, for your written assignment. And those of you that have not are going to find out that a lot of them have already been taken. So the more that have been taken, the more remote and the more rare the chemical messenger becomes, and the harder it is for you to find research on it. So that's something to be thinking about when you're trying to organize the last half of this semester. You want to make sure that you've got your topic chosen so that you can spend some time researching it. So with the nervous system, Sarah, you've got your hand up. Just use your microphone. Just turn on the mic and ask.
Yeah, sure. Sorry, Dr. Anderson, I emailed you about my assignments subject, but I didn't receive your feedback. So I don't know.
Okay, this is not for, this is not for right now to talk about this, but I do recognize that you emailed me, and I'm sure that I sent you back a response, but I'll double check when class is finished.
Sure.
And if for some reason I don't, then just resend me that email.
Okay, sure.
Okay, just to be sure, for sure. All right, so with the nervous system uses electricity to communicate. And as we can appreciate, electricity is going to be fast. We've already talked a little bit about how that electricity works when we looked how we signaled a muscle cell to contract between a communication between a neuron and a muscle. So in the nervous system, it's very similar to that. There's going to be a excitation and there's going to be a depolarization, and those things are going to promote the passage of a signal throughout the nervous system. So we're going to look at the components of the nervous system, how the nervous system is organized. So it's very fast because of this electricity pathway. The endocrine system is much slower because it uses blood primarily as the mode of transportation. And we can appreciate that blood flows much slower than electricity travels. So the chemical messengers will arrive at their targets later, but typically their effects are longer lasting. So when you think about the nervous system, it's fast and short-lived. It's electricity, like flipping on a switch and then flipping it back off again. So we generally organize the nervous system into a central nervous system, which is basically the brain and the spinal cord. And the peripheral nervous system is everything else. So that's going to include nerves and receptors. And we're going to put in effectors here as well. Now, effectors technically are the destination for the nervous system signal. So technically the effectors are outside of the nervous system, but they are connected to the nervous system. So we'll include them here. Okay, and we can do a little schematic that really simplifies the entire nervous system. And we can categorize or compartmentalize, if you like, the entire nervous system into 3 boxes and 2 pathways. So the boxes would look like this. And we would have a pathway going to another box and another pathway out to another box. And this little line diagram here completely describes the entire nervous system. All we have to do is fill in a few things. And then as we learn a little more about the nervous system, we can add to our little diagram here, but it still remains a very effective description of all the components of the nervous system. So what we would do is we would divide with a horizontal line so that everything below this dashed line we just drew is peripheral nervous system. And everything above that line is the central nervous system. Okay, and we can divide the nervous system a little further vertically so that everything on this side would be considered sensory, and everything on this side would be considered motor. In the nervous system, in biological systems generally, if you use the word motor, it means that the tissues are active. They do something. So we've already talked quite a bit about muscle tissue. We know muscle tissue contracts. That's a motor activity. So we talked briefly about smooth muscle and cardiac muscle, and we talked mostly about skeletal muscle, but those are 3 examples of motor tissues. Glands that secrete would be another example, and adipose tissue is a 4th example of a motor tissue. So those are going to be part of our schematic here that we're looking at. So in our first box, we're going to put receptors. So a couple of things about receptors that's important to remember. So when we think about receptors, receptors in the nervous system, but in the whole body generally, are described as being specific. And you're going to find out about this because your chemical messengers will only respond or cause change on cells or tissues that have receptors for them. So they're specific. So if there's a receptor for example, insulin in the body, a receptor for another hormone, say growth hormone or thyroid hormone, would not respond to insulin because it's specific. It's like a lock and a key. The receptor will be specific to the signal molecule. So there's specificity. That's an important concept to remember. And receptors can fall into 3 general categories, really 2 main categories. They could be described as extero receptors, meaning they're going to respond to external stimuli, like the temperature in the environment, or touch, or pressure, or what's another one that they might, pain, for example. All things coming from the external environment, as opposed to intero receptors. And interoreceptors can respond to a lot of those same things, but they're receptors that are found on the organs internally. So maybe on the cells associated with the stomach or the lungs or the heart. And they'll measure things like temperature and chemicals in the blood, for example, in order for the nervous system to be able to be exposed to those things and initiate changes if necessary in order to maintain homeostasis. And a third category that sort of crosses between the two are called proprioceptors. And those are receptors that are found in the ligaments and in the tendons around joints. And what they do, and so they're internal receptors, right? They would be classified as internal because they're inside, but they respond to length and tension in the muscles and tendons, which is usually a result of things that are happening outside the body, movement, running, standing, keeping your balance, those sorts of things. So they're really measuring changes in the environment, external environment, with receptors that are in the internal. That's why they sort of cross over between the two. Sometimes they're considered a third category. Doesn't really matter, but if you think about proprioceptors, they respond and adjust to body position. And the extro and intro receptors, they can respond to these other sensations that I mentioned already. Sensations like touch and temperature and pain. We okay with receptors?
Yes, sir.
Okay, so if we're going to take the information that these receptors are receiving and introduce it to an integration center, in other words, a group of cells that can do something about those sensory stimuli, we're going to have to introduce it to the central nervous system. In the nervous system, integration occurs in the central nervous system. So this integration is going to occur in the central nervous system, the brain and the spinal cord. So very complex integrations are going to occur in the brain. Very simple integrations can occur in the spinal cord. So a spinal reflex is an example of a complete illustration of all of these boxes and all of these pathways, but very simple. If you think about that knee-jerk reflex, if you've ever had a knee-jerk reflex performed on you, or you know generally what it is I'm talking about, it's an example of a very simple type of integration. It went into the spinal cord and back out. There was no requirement to go all the way up to the brain and integrate in any kind of complex way. But if integration is going to occur, it's going to occur in either the brain or the spinal cord. And then when integration has been completed, we're going to send out a message on the motor side to realize a response. So ultimately what we're going to see is on the motor side, we're going to finish our pathway on effectors. And effectors can be described as somatic or autonomic. And if it's somatic, we already know what the answer is for somatic. That's skeletal muscle. So we know a lot about skeletal muscle already. We know that for contraction to occur, we have to excite that muscle. That's the responsibility of the nervous system. If we rest our hand on a hot burner on the stove, we're going to want to remove it very quickly. And to remove it, we need skeletal muscle. So the stimulus would be pain. And we would come up and we would integrate very quickly and come back down and lift our hand away from that source. Complete integration, and utilization of the entire nervous system on that very quick reflex response to pain. Okay, on the autonomic. Oh, sorry, question?
I was just wondering, what are like the characteristics of the integration again?
So integration, well, listen, we're going to spend the next 3 weeks talking about integration, but that's where the information from the receptors is going to be received, and what's going to be determined is whether it's too high or too low as it's approaching the limits towards homeostasis, and adjustments are going to be made.
Okay, thank you, sorry.
Okay, so when we go down to the autonomic, all the other tissues that have the potential to do something, in other words, have a motor responsibility, would be smooth muscle. And smooth muscle is found in hollow organs like the stomach and the intestines and blood vessels all over the body. Cardiac muscle. We know cardiac muscle is going to be found associated with the heart. And glands. And adipose tissue. So we really have 4 targets for the autonomic division of the motor pathway, and 1 on the somatic side, skeletal muscle. Remember, we talked about skeletal muscles under voluntary control or conscious control. So it stands alone. The autonomic effectors are going to fall under subconscious commands. So our heart beats at 60 to 90 beats a minute without us having to think about it. We breathe in and out, 14 to 16 times a minute without having to think about it. Glands secrete to adjust for body temperature or those sorts of things without us having to think about it. Right? So when we think about these effectors, we already know a little bit about them because we've been kind of talking about some of this stuff already. We know a bit about glands. We know about sweat glands and oil glands, for example. We know a lot or a fair bit now about skeletal muscle and the way that it operates. So we can see that effectors are motor and receptors are sensory. And integration is going to have components of both sensory and effector depending on where in the integration process we happen to be. But the point with integration, because it was a good question that was brought up, is to understand that we're taking all of this sensory information and we're inputting it to the brain and the spinal cord. And the goal of this input is to come up with an appropriate and efficient response. So think about what it's like to drive a car. When you drive a car and your hands are on the wheel and you got your Tim Hortons coffee in your coffee mug and you're going to take a sip, you grab the cup of coffee, it's warm, you bring it up to your mouth, you can taste it. So those are all sensory types of experiences. You've got your hand on the steering wheel, you have your foot on the gas pedal, and then everything is homeostatic. You're enjoying your coffee, you're driving in a straight line, then somebody cuts you off. Well, in the flash of a second, you've got to jam your foot on the brake, you've got to throw the coffee away or put it back in its place, and you've got to respond to that change in the environment. All of that sensory input, visual, sound, touch, which would be called somatic sensory, anything that has to do with the receptors in your skin, like touch and temperature and pain, would be described as a somatic sensation, because soma is just a Greek word for body. So in this case, if it's somatic, it has to do with your body, and specifically the outside of your body. So we've got all of that sensory information coming in. So we can look at this. Let me show you another sort of way to look at the way that this system is organized. So remember we said that the brain and the spinal cord. So we're going to be talking about the actual organization of these structures. We'll probably get started on it later today. But what we've drawn in turquoise then is the central nervous system. We're looking at a lateral view of the shape of the brain and the spinal cord. We've got the cervical curve, the thoracic curve, and a lumbar curve drawn in here. Remember, the spinal column protected the spinal cord. The skull protects the brain. So we can have, I can't remember what colors we had, but we can have sensory information coming in from all kinds of places. So we would have our receptors. Coming in at various levels of the central nervous system, and they're going to travel up. If they enter the spinal cord, they're going to travel up the spinal cord and into the brain for integration. So what I've drawn in yellow, except for that central stuff, actually, let's be consistent here. So once we get to the spinal cord, by definition, we're dealing with central nervous system pathways. So I'm going to draw it in a different color because it's in the central nervous system. So our yellow is peripheral nervous system. Now, once we've received all of this sensor information from all over the place, let's say this one, the lower one here, is sensation of touch your foot to the gas pedal. And this is sensation from your stomach because of the coffee and the sugar that's in the coffee. And then we've got movements of the neck and movements like visual and maybe hearing coming from these different levels of receptors. And we're going to input to the central nervous system for integration, and then we're going to output. And we're going to output, we're going to come down through the spinal cord, and we're going to leave at different levels of the spinal cord. And we're going to finish on effectors. So green and yellow in our little scheme here represent the peripheral nervous system. Those arrows are nerves. And the motor arrows are finishing on effectors. And the sensory arrows came from receptors. Does that make sense?
Yes.
Yes.
Okay. So a nice clean picture of this can be looked at right here. And when we look at this picture, we can fill it in very quickly. As soon as I get the right slide up here. So we're going to put in our receptors. And our receptors fall into categories like external and internal, right? And we can also describe them as being thermoreceptors. What do you think thermoreceptors respond to?
Heat.
Or?
Temperature.
Oh.
Yep, heat or cold, temperature. Temperature is the best way to summarize that. Now, we also can categorize some receptors are described as mechanoreceptors. Mechanoreceptors will respond to things that are mechanical. Touch would be an example of mechanical. Touch and pressure, those sorts of things are mechanical. We've got chemo. What do you think chemoreceptors respond to?
Chemicals.
Sure. So blood glucose could be included there. Carbon dioxide or carbon monoxide could be included there. Oxygen would be included there. The levels of sodium and potassium would all be considered chemoreceptors. And all of those, this blue side represents sensory in our little scheme here. So we're going to send this information in a pathway that's usually nerves, if it's in the peripheral nervous system, and we just call them tracks. Tracks are spelled with a second T, tracks. So the pathways in the central nervous system are tracts, and they're called nerves when they're in the peripheral side. And then we get to the brain and the spinal cord. Central nervous system, that's where integration is going to occur. So this, again, will be a tract. So we can see with the way we drew this arrow, and this will become more evident as we build on what we're talking about. But you can look at this picture, and I could ask you questions about the nervous system. For example, I could say, if information is traveling in an ascending direction, in other words, it's traveling upwards in the nervous system, what sort of information will be carried in an upward pathway? We got the arrow here. If we're traveling towards the brain and spinal cord, what kind of information are we carrying?
It can be thermal or mechano or chemo.
It would be, and
Sensory.
all of it would be categorized as sensory, right? So for that kind of question, it's very basic. Sensory. If it's carrying away from the brain and spinal cord, and would otherwise be described as descending, what kind of information is it going to be carrying? If it's not sensory, it's
mechanical.
Motor.
motor. Which means it's going to finish on an effector that has action. Skeletal muscle on the somatic side, smooth muscle, cardiac muscle. By now you should be able to say it along with me, glands and adipose tissue. So when we look at this picture, we have the entire nervous system represented. And if you can look at this picture today and say, yeah, I know exactly what he's talking about. I can talk about the nervous system for a long time just by looking at this diagram, then guess what? You don't need any more lecture on the nervous system, because if you can look at this and understand what it means, you've got the whole nervous system figured out. Okay, so this describes the way the nervous system is organized. It's very, very functionally organized. On this side, everything on this side is sensory. Everything on this side is motor. Everything below that dashed line is peripheral. And everything above the dashed line is central. Central is brain and spinal cord with these tracks. And this little diagram that we spent a few minutes on is exactly the same diagram, but we've just added in a little bit of anatomy into this, but it's the same drawing. We've got, we can our yellow box and when we get above the line where we're entering into the peripheral nervous system, I think I used this color for the brain and spinal cord, and we'll just use that color there. So this is the same, drawing the same thing two different ways. One might work better for you, whichever one works better for you. But all the components of the nervous system are represented here. So you should be able to, you can make up your own mind map, one that works for you, but you should be able to talk a little bit about receptors, a little bit about integration, a little bit about effectors and their pathways if you're asked to talk about the way that the nervous system is organized. We okay with that so far?
Yes.
I have a question.
Yeah.
You said like it's carried like ascending or upward is sensory, right? And it's like descending, it's motor.
Right.
What is being carried? I didn't get that. Can you
So
repeat,
that's
please?
it. So remember, so that's a good question because we didn't really talk about that. But the message that comes to a receptor, a specific receptor for a specific stimulus, is what's going to be carried in and up for integration. So it might be the signal for pain. It might be the signal for touch. It might be the signal for temperature. And it's going to be traveled and it has to be converted to an electrical signal. So that's where your question is really important, and we're going to get there. We're going to talk about that. But so a physical stimulus has to be converted to an electrical one in order for integration to occur. So the pain that you receive when somebody pinches you or you cut yourself with a knife or something like that has to be converted from being a physical injury to an electrical one. And it'll be carried by specific receptors and specific fibers, neurons, that carry pain up to the brain to say, where did that come from and how bad is it? And then we'll come back out and decide what we have to do. But that's all by electricity. So it all happens really, really fast. And we're going to talk about that electricity shortly, very shortly. Are we okay? Is that, for now, does that help you?
Yeah, thank you.
Okay, good. There are good questions that are coming out today. Now, why am I not getting this to clear? There we go. Okay, so in order for this pathway, now let's just draw our little pathway. 3 boxes. And if you haven't figured out by now that this pathway is, this little schematic is kind of important, you probably should be thinking along those lines by now. So when we look at this pathway, we realize that, okay, we get the responsibilities now. We've got receptors, we've got sensory motor, we've got integration, we've got effectors, central peripheral. We sort of get the organization, but we don't, like the question that was just asked about what exactly is being carried in there. Well, that's a great question because we haven't really talked about that yet, but that's what we have to talk about. So what is it that's going to make this work? And it's these cells that are part of this system. Like always, it was the cells that were responsible for contraction in the muscular system. It was the cells that gave our skin the characteristics that our skin has. The outer layers are dead cells. The inner layers are replacing those dead cells on a regular basis. The characteristics of those cells are pretty unique. Neurons are pretty unique all in their own. So the one characteristic that makes neurons really unique, and only muscle cells have this same characteristic, and that's excitability. So when a neuron has excitability, it means that its membrane has a resting potential. Okay? Which means it's polarized, just like we talked about when we talked about muscle cells, and it's more negative inside. And that was simply because we have chloride ions inside and we have some proteins inside that are negative to a greater degree than on the outside, and we have a lot of positively charged ions on the outside that make the inside of a cell, especially an excitable cell, more negative than on the outside. So that's where that cell becomes polarized. And if we depolarize to a threshold, we get our action potential, right? This is a bit of review. We're okay with this so far, right?
Yes.
Okay. And if we hyper polarize that membrane, then we would not get an action potential. Right? So I don't like using the numbers because some people get excited about that. But if we said that our resting potential was about 70 millivolts, okay, now nobody should bother memorizing this. If you've taken some of this before and you know these numbers, that's just fine, and you can use them as examples. But you do not need to memorize them. What I want you to do is get the concept, get the idea of what's happening. If we depolarize, we're going to become less different. We depolarized. So we're going to go towards zero. In other words, we're going to add positive ions to the inside of that membrane. So our threshold is about a minus 55 millivolt. If we get to a minus 55 millivolt, then what we're going to do is we're going to shoot right up into the positive zone. So if we were looking at a graph, we would say, here's our minus 70. This is our cell at rest, minus 70. If we dropped enough positive ions inside the cell to take it up to threshold, then boom, once we get to threshold, we're going to see, and it might, let me redraw that a little bit. I just want to show you something. So what we'll do is we'll say threshold. We could even go, this could flux a little bit till we get to threshold. Once we get to threshold, we're going to go all or nothing. In other words, this depolarization, the floodgates open and positive ions from all over the place near the location of where this is all happening open up, and we get this huge influx before it starts to come back down again. And actually what I'll do, and then when we start to come back down, anybody know what the term is when we start to come back down?
Repolarization.
Repolarization, right?
Repolarization.
Yep, repolarization, and then we're going to reset. Okay, so basically, if we go more negative, if you notice in the process of resetting, we actually dropped a little bit below, let's say we're at minus 80 there, just for examples, okay? So during that little dip where we're below minus 70, we're hyperpolarized. We're further away from an action potential. So that's giving the cell, in this case the excitable cell, time to reset itself, reorganize. So these are electrolytes flowing in and flowing out across the membrane that cause all of this to happen. In an excitable cell, this area right here is our action potential. And in a skeletal muscle cell, we know that if we get an action potential in a skeletal muscle cell, we get contraction. If we get an action potential in cardiac muscle or smooth muscle, we get a contraction. If we get an action potential to a gland, it's going to produce. You get what I'm saying there as far as the purpose of that action potential? It's a signal. It's an electric signal. We okay with that?
Yes, sorry, I do have one question though. How have like doctors or scientists measured this to like an approximate number? Like, is there physics to this? We should know that. Well, I'm just curious.
No, first of all, no, you don't need to know it. It's very complicated. It's really amazing what some of these guys have been able to do with really primitive equipment. But because it's electrical, it can be measured with with tools that measure electrical changes, electrical activities. So it gets measured, we can measure brain activity with an electroencephalogram. It literally measures electrical activity in the brain. They've done this. It's the same concept to measure individual cells' electricity. So they determined that these cells have a charge across their membrane that represents a difference of about 70 millivolts. It's 70 millivolts more positive outside than inside. The other way to look at that is 70 millivolts, I should say, less inside. So it's negative 70 millivolts inside, more negative inside than outside. So those are measured by voltage meters. And then when they can literally experiment by stimulating a change in that polarity. And they discovered that if they put the equivalent of positive ions to a threshold, which in our example here was minus 55, as soon as you hit that threshold, then it created this all or nothing response. It's an example of positive feedback. So a little bit of positive influx results in a complete depolarization of the whole membrane of the cell. So this whole process might start. So here's our lipid bilayer of a cell, and it's stimulated by a neuron, okay, just like our muscle cell was. So the process of influx starts here, right? When the neurotransmitter released and bound, it opened up channels, starts here. But if that's enough to get to threshold, then it's going to open up channels all the way along, and we're going to get positive ions depolarizing all the way along this membrane in milliseconds, all or nothing. So this whole cell gets excited, and if it's a neuron, what it's going to do is transmit that electricity along a pathway. I'm going to show you that when we look at what a neuron looks like. Okay? So that's measured by voltage meters. They can literally measure the change in voltage activity across this membrane.
Cool, thank you.
Yeah, and I mean, there's a lot more to it than that. And a lot of these primitive, and I don't mean primitive, older experiments, a lot of times they did them on themselves because it really wasn't very, moral to be trying to get volunteers to be subjected to electrical shocks and electrical stimulation. So a lot of times they did their own experiments on their own cells to try to get some of this information. When we talk more about the brain, we'll find out one thing that's sort of not necessarily very intuitive, but the brain has no actual receptors on it. So if you opened up the skull and poked the brain, you won't feel it, and it's not painful. But you can observe electrical activity. You can put probes on the brain and measure and monitor electrical activity. So if you're trying to follow a pathway, let's say you put a pain stimulus on your baby finger on your right hand. Well, it's going to travel along the finger, along the arm, through the shoulder and back to the spinal cord. And from the spinal cord, it's going to travel up to the brain. And that can be measured all the way along. And you can skip if you want. If you poke the baby finger and you look at the activity in the brain, you can see where it finishes on the brain. What part of the brain will monitor that exact stimulus in that exact location? In other words, when we talk about the brain, we'll find out that we have neurons in the brain for every area of the body. Groups of neurons that are responsible for sensation of that baby finger on the right hand. And they're predictable and measurable depending on what signal you're sending to them. So it is very fascinating and very complicated too. At one level, it's simple, but to actually discover it and measure it and all that kind of stuff, it gets very complex very quickly. So these neurons that we're talking about, they have excitability, which means they can generate action potentials. And that means they're going to be able to continue an electrical signal through the pathway that we drew up here, from a receptor into the spinal cord, to the brain, out of the spinal cord, and to an effector. Repeat, repeat, repeat. Could be tens to hundreds to millions of neurons involved in the pathway. But the concept all the way along is going to be the same. We need to generate an action potential in each neuron that's part of that pathway. And we'll be talking about that some more in a few minutes. But so that's the one property of the neuron that we want to remember. It's excitability. What else? So we'll call that property number 1. The other thing about neurons, which is kind of unique, you wouldn't expect it, is well, this part you probably requires lots of ATP. Neurons are busy. And by busy, I mean they need a lot of energy. So they need a constant supply of a lot of ATP. And they probably utilize, anywhere from 20 to 25% of the body's entire blood flow and oxygen delivery to the neurons. So in other words, if you've got 5 liters of blood at any given time, 1 liter of that blood is going to be up in the brain, up in the central nervous system, supplying oxygen to the neurons. They need a constant supply. Okay, and they really are not very good at performing glycolysis. Remember, glycolysis produced a little bit of ATP, and we didn't need oxygen. But for these neurons, it has to be aerobic. So they need a constant supply of oxygen, and they need a constant supply of glucose. And they cannot store. Remember, skeletal muscles could store both, but neurons can't. So this is why I'm pretty sure I mentioned it in class. The first group of cells in the body that are going to react to a lack of oxygen are your neurons. They're going to get very excited very early. Within seconds, certainly within a minute or so, they're in an emergency state because if their oxygen supply was interrupted. Because the only way that they can make enough ATP to perform and survive is if they get a constant blood flow. So that's why I say 20 to 25% of blood flow, and that's at rest. Never mind when demand goes up. So we need 20 to 25% of the total body's blood flow to be passing through the brain and the spinal cord in order to supply this oxygen and glucose. Does that make sense? We okay with that?
Yes.
So those are big things to remember about neurons. Now, when we classify neurons we know already they're excitable. And they don't have centrioles. And some of you might remember from your cell that if you don't have centrioles, you typically can't undergo mitosis. So mature neurons, if they die for any reason, they can't be replaced because they're not undergoing mitosis like skin cells do. Now, there are a couple of areas in the brain that have some stem cells that we can call upon under the right circumstances. But a lot of neurons in the body, a lot of neurons in the brain and spinal cord, once they've been damaged, injured, or killed, they can't be replaced. So it'll be a loss of function by definition. So that's another thing to keep in mind about these neurons. Once they're mature, typically they do not undergo mitosis. So there's kind of a fixed number, more or less. So neurons can be classified by form or by function. What does that sound a lot like? Form and function sounds a lot like?
Muscles.
Like bones.
No. Like anatomy and physiology. right? Muscles and bones have those features too. But more generally, if it's form, it's shape, right? It's size, shape, that's anatomy. And function is physiology. So when we classify neurons, they can be classified as unipolar, bipolar, and like most things in anatomy and physiology, once we get past 2, we're going to call it, in this case, multipolar. And that's looking at shape. Okay, so unipolar, we're going to have a cell body and a nucleus. And that cell body is probably going to have some specialized features associated with it that help it to produce neurotransmitters. What's an example of a neurotransmitter that we talked about before the break?
Insulin?
No, insulin's a hormone.
Oh,
Acetylcholine.
But, yeah, acetylcholine was the example that we used. That was the one that stimulated the skeletal muscle to contract. So insulin is a chemical messenger, but it's part of the endocrine system. Now, if it's unipolar, it means that there's one pole that's coming in and out of that. So it might come from multiple locations, and it might go to multiple locations, but at the cell body, there's just one post, unipolar. So now we got a good idea what we're going to see in the bipolar. We're going to have one pole that leaves the cell and one pole that enters the cell. And if it's multipolar, we're going to see that there's typically just going to be one pole that leaves, but there might be hundreds of contributions to this cell membrane, this cell body here. So that's multipolar. So each of these poles, when we look at the poles, like we might have a receptor on this side, and we might have several receptors or other neurons in this pathway. But you can see that if there's a whole bunch of receptors, there's a whole bunch of stimuli being funneled in to the body of this particular neuron. Okay, so that's simply what these neurons look like. So unipolar are examples of unipolar neurons are found in the peripheral nervous system that are described as somatic sensory. So now, if you've got that word sensory in there, we're talking about their function. These are somatic sensory neurons. So that's an example. We're going to, don't worry, you'll find out where these are found as we move along in the nervous system. Bipolar neurons are often associated with special sense. So we're going to see some bipolar neurons associated with vision and with smell. Those are some examples of where we would find bipolar neurons. So special sense is different than somatic sense. Somatic sense is taking information like touch and temperature and pain and delivering it to the central nervous system. Special senses are the other senses we think about, vision and smell and hearing and balance. Those types of things are special senses. And most motor neurons fall into this category of multipolar. So when we're looking at motor or special sense or somatic sense, those are all effects. Those are all functions. So they would be described as the function or the physiology part of these neurons. Okay, now these are just some examples. But now in terms of uni, bi, and multi, those are the most common organizations of the way these neurons work. Okay, so we can classify them by their function, sensory or motor, or we can classify them by what they look like. One pole going into the cell body, two poles, multi. And in all these cases, the pole that leaves and carries the electrical signal away from the cell body is called an axon. So we can see here that no matter how many poles these neurons might have, they are going to have a single axon carrying the signal away from the cell body. In other words, on the other end of this axon, here and here and here, there's either going to be another neuron or another effector, like our skeletal muscle, our smooth muscle, and so on. And the pole or the pathway that carries the electrical signal to the cell body, anybody know what that's called? That's going to be called a dendrite. So the dendrites, and we have a lot of dendrites down here. Those would all be called dendrites. Okay, so the electric current in a dendrite is being carried to the cell body of that neuron, and the electric current in the axon is carried away from the cell body. So that's what's going to continue on. If we looked at a receptor that might be connected to a dendrite that's going to be connected to a cell body that's going to be connected to a second neuron, now we have a pathway. And this was our first neuron. That's our second neuron. And we're showing 3 different neurons. But this neuron could have inputs from other neurons. And now you can start to see how it gets complicated. So what did we call the neurotransmitter? Sorry, I just answered the question. The chemical that gets released at one of these connections, these synapses that we're talking about right here, and right here is a synapse, and there is a couple of synapses in that picture too. But it's an electrical signal that comes to the axonal ending, and we know that to continue the signal, we have to release a chemical. And the chemical was called a neurotransmitter. The neurotransmitter that we know a fair bit about already is acetylcholine. So when acetylcholine was released, and it was released onto a skeletal muscle, when ACH was released onto the skeletal muscle, what happened to the skeletal muscle?
It contracts.
Yes. If you excite muscle, only one thing's going to happen. Contraction.
Sir.
Yep.
I just have a question. You wrote dendrite. Is that like the I want to say almost the body of the neuron on the left side, or is that like the receptors?
No. So here, a receptor is a separate entity. Okay. And it has to connect to a dendrite to carry the first action potential to the first neuron. So that's what has to happen here. We have to have an action potential that's generated, for example, by the signal pain, that's going to be received by our first neuron in the pathway. And that neuron is going to synapse on a second neuron, for example, in the spinal cord, where we're going to have to release a neurotransmitter here, right? The action potential got generated, traveled along, we transferred through a neurotransmitter, generate a new action potential, neurotransmitter, a new action potential, and the neurotransmitter that finishes to excite a muscle was ACH. Yeah?
Okay, thank you.
Okay, so we're gonna talk a lot more still, partly as a review of what the steps are that are involved with that process there, but that's where we have to get started. So again, it all comes back to this. Right? These were our receptors. These are our effectors. So we're going to receive a sensation. Maybe it's pain, maybe it's pressure or touch, and we're ultimately going to have a response. The response will be manifested in the effectors, and the receptors will initiate the stimulus to an electrical signal, the way we've kind of drawn it in here. Okay, are most people with me on this? You understand what I'm saying?
Yeah.
Okay, so if we had, let's say we had a receptor, and let's say it's a pain receptor, just because most of us can kind of relate. So what happens during a pain signal is cells are injured, they're broken, and they release chemicals. And the chemicals are going to get picked up by these nerve endings that are nearby the cells. So we're going to draw a couple of nerve endings in here, and we're going to have them join up and travel to a nerve. A neuron. Here's our first neuron in our pathway. But we started with our receptor, and then that neuron is going to synapse with a second neuron. And this one is happening, this one is peripheral. This one's going to happen in the spinal cord, and then it's going to travel up the spinal cord and synapse, let's go with this one, on a third neuron. And then that one goes right up into the brain to those neurons that I talked about before. So if we put a little tack and we prick the end of our baby finger on our right hand, that's what's going to be monitored right here at the pain site. Then we're going to take that pain signal and we're going to enter the spinal cord, synapse with a second neuron, and travel up the spinal cord, synapse in the brain with a third neuron, to finish on a group of neurons in the brain that are dedicated to receiving information from the baby finger of the right hand. So now we're up in the brain. So in the picture that we drew before, where we had our spinal cord, okay, so we can draw this exact same thing. So here's our baby finger. I'm going to draw a big baby finger. Right, here's another finger, another finger. Okay, so what we did is we took a thumbtack and we stuck it into our baby finger. So the nerve endings that are near there are going to travel towards the spinal cord, but outside the spinal cord, they're going to travel to a neuron that's outside the spinal cord that enters the spinal cord. Okay, so that's where this orange pathway goes. Now we're on to a second neuron, and we're going to travel up to a third neuron. What color was our third neuron? Turquoise. We're going to finish there, and then we're going to go up to the purple neuron in our pathway. So we just drew the same thing two different ways, right? You follow me on that?
Yes.
Okay, so here's the thing. At every communication between a neuron and the next one, we have to have an excitation, a electrochemical transfer. So there's got to be an electrical chemical transfer all the way along at each of those junctions if we're going to continue the signal. So that looks a lot like what we've already talked a lot about when we looked at the musculoskeletal junction. The neuromuscular junction. But in this case, we're in the nervous system, so we might be looking at the communication between two neurons in the same pathway. So what we've now done is taken a look at this. We're just going to look at a bigger version of what's going on between those two neurons right there. Okay, so we've got neurotransmitters here. And we're going to have receptors for those neurotransmitters on the membrane of this neuron. So we can think of this as neuron number 1, and this one is neuron number 2. And we know that inside this neuron, it's going to be polarized. It's more negative inside than outside. So when we delivered our pain signal, and the chemicals that stimulated those nerve endings generated the first action potential. So the action potential a pain signal gets converted to an electrical signal at the site of pain, and the action potential is going to travel like electricity all down that membrane till it reaches the axonal ending. And just like we saw with skeletal muscle, there's these calcium channels here. And when the electricity reaches these channels, they're called voltage-gated channels, they're going to open up, and calcium gets to rush in. And calcium, with its positive charge, is going to stimulate exocytosis of the neurotransmitter. So now this neurotransmitter, we're not even going to bother naming it, but if we wanted to, for example, ACH, and ACH is going to get released, or a neurotransmitter is going to get released, and if the receptors are specific, and we get enough of them, what's going to happen? The neurotransmitters bind with their receptors, and if we're going to get a new action potential in the next neuron, what's going to happen when this neurotransmitter binds with its receptor? What happened in the muscle cell? Same thing here.
Contraction.
Well, no. That's not yet.
Is it a reflex?
No. What happened when ACH bound to its receptors on the muscle cell? What happened next?
Oh, is it pain?
No. We didn't talk about pain when we talked about muscles.
Would it be the ion channels open
Yeah, for what? Which ion?
for sodium?
Yeah. So those sodium channels opened up, and we had lots of sodium outside. So as soon as we open up channels, we go downhill. And sodium gets to rush in these open channels. And if we rush enough sodium in that we reach threshold, what's going to happen? Well, we talked about it. Not only did we talk about that when we talked about the skeletal muscle, we just talked about it about half an hour ago in this class.
They reset?
No, what's going to have to happen first? We reach threshold, then what happens? What's the purpose of reaching threshold?
For action potential?
All or nothing, right? The whole membrane is going to switch because sodium channels all the way along the length of this membrane will open up and rush in, will depolarize the entire membrane, which means we're going to get a new action potential that will travel down the whole length of that membrane to the next synapse. And guess what happens at the next synapse? Let's see here. They were at the next synapse, we got an axonal ending with neurotransmitter in it. and it was synapsing with the third neuron. And it's more negative inside than outside. Now we have a new action potential that's traveling. Electricity, voltage arrives at the axonal ending. What's going to open up?
Ion channels.
For,
Calcium.
there we go. And when calcium rushes inside that axonal ending, what does it stimulate?
The neurotransmitter.
What about it?
ACH. It wants to come inside.
Right, exocytosis. And it's going to bind on its receptors, and enough neurotransmitter binds to those receptors, then what's going to happen?
Sodium channels open up.
Yeah, there we go. Sequence of events, right? One before the other. Just like we talked about in muscle. The beauty of this excitation reaction, the electrical and then the chemical messaging, is it's a repeat. We saw this the same way when we talked about how a muscle cell contracts. The difference is the new neuron is not going to contract because neurons don't contract, but it will generate a new action potential to travel along its pathway until we get an effector response. So we're going to travel ascending with the sensory information, and then we're going to travel descending with the motor information. And guess what our response is probably going to be to this particular example right here? What would you predict would be the response?
Contraction.
Oh, well, careful. More general than that. Don't get too specific.
They're gonna reflex like pull away.
Yeah, you're going to pull away. That thing hurts. So you're going to try to withdraw, right? So will it finish on skeletal muscle? Yes. And will we get contraction? Yes. But let's just look at the big picture first. So this would be an example of a withdrawal reflex. We withdraw from the pain source. But you can see that what we did is we started with a receptor. We carried the sensory information up to integration, the brain and spinal cord, and back out again to effectors. In this case, we started with pain and we finished with withdrawal, right? Withdrawal. Does that make sense? Do you follow that?
Yes. Can you like repeat the steps for the action potential one more time? Like how synapses goes?
Yeah.
Thank you.
Okay, so this is the way we have to go. Okay, so we'll let's put our picture over here. This is our presynaptic neuron. Okay, and this one is our post synaptic neuron. Right? We okay with that terminology?
Yes.
Okay, and we know it's more negative inside than outside, right? It's resting at our -70, for example. Okay, and we've got these vesicles of neurotransmitter that are sitting in the axonal ending. And when electricity, in other words, the action potential, travels down this membrane, so this is our action potential, traveling down that membrane, gets to the axonal ending, it's at the end. But what's waiting at the end are some voltage-gated channels that are closed until that electrical signal shows up, and then it opens up for calcium. Whoops. And now calcium rushes in from lots of places because calcium is relatively high in the extracellular fluid and low in the intracellular, so there's a downward grade. Okay, that's going to initiate exocytosis. We're going to write all this down. Okay, and the exocytosis results in our neurotransmitter being released into that synaptic space. And if it binds to its receptors, it's going to, they're typically connected to closed sodium channels. That now can be opened up. And when they're opened up, sodium that's on the outside can flow downhill and get inside. And if enough sodium rushes inside that we go to threshold, remember when we drew that threshold was when we got up into this number right here. If we got to threshold, we got all or nothing, which meant we got an action potential that traveled through the entire membrane of that cell. So if that cell looks like this, and we generate the action potential up here, it's going to travel all the way down to the axonal ending the way we've drawn it here. So we've just repeated the whole thing. Now we start in the next neuron. So it's electrical to here, electrical, and then it's chemical, the neurotransmitter, and then another electrical if we got to threshold and we got a new action potential. Boom, 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 all the way through the nervous system till we finish on an effector. So our first action potential is generated from a receptor and dendrite of the first neuron. Okay, so that's what we've got right here. If we said this is number 1 and this is number 2. So we started with our receptor. Let's say it was pain, right? So our pain signal from our tap, right? All I've done is taken the finger out of the picture. We okay with that?
Yes.
Okay. So the action potential travels the length of the first neuron. So dendrite to cell body to axon. Where the voltage gated Okay, everybody with me so far? Can you read my writing?
Yes, sir.
Yeah. So we're up to the point where we've got our neurotransmitter being delivered outside of the axon, right? That's where we are right here at number 4. We good?
Yes. But I have a
Excuse
question.
me.
Go ahead.
Yeah, can you just read number 4?
Calcium inside the axon stimulates exocytosis of the neurotransmitter. Right? Our voltage gated channels for calcium opened up when the action potential arrived at the axonal ending. Yeah?
Yeah, thanks.
Okay. So now we've got the neurotransmitter binds to receptors on the postsynaptic neuron. Which then allows sodium
Sorry, what's the word after sodium?
in number 6.
Yeah.
Rushes. Sodium rushes inside. Does that work?
Yes, thank you.
Okay, so now if enough sodium rushes in to reach threshold, and not a number, but just to remind yourself of what we said threshold was in that previous slide earlier today, we get to that threshold, we get all or nothing. Which is a new action potential. And those steps will repeat themselves at every synapse, and there could be millions of them.
Sir, just a question. Where does ACH fit in this?
So ACH is an example of a neurotransmitter. Okay, so ACH would, in the neuromuscular junction, we could put ACH here in the place of a neurotransmitter because it's one example of a neurotransmitter. But
Okay,
there's probably over 100 neurotransmitters out there. You know, some we know exist. We don't even know what they look like just yet. It's a
Sir,
little
can
bit.
you go to the first step?
Step number 1.
Yeah. I just wanted to see it.
Say it again.
I just wanted to see step number 1.
Oh, you mean as long as you can see it?
Yeah.
So that's how we have to get started, right? And we know from our 3 boxes and 2 arrows, we ought to start with a receptor. So our receptor has a responsibility, essentially, to convert a stimulus, whether it's mechanical or chemical, to an electrical signal, in other words, an action potential to get things started.
Okay, thank you.
For number 5, is that covers or carries NA channels to open?
Pardon me?
For number 5, step 5, is that like covers or carries NA channels to open?
Causes sodium channels to open.
Okay, thank you.
I can't believe you don't know how to read my writing. And of course, I'm just kidding on that. My
I
writing.
tried.
Yeah, you I think you had it, but I can definitely understand why you might have some trouble with my writing. So never hesitate to double check what I've said and make sure that what I've said and what I've spelt are the same thing. Okay, so wherever we happen to be in the nervous system, we're going to see this exchange going on. So we could super simplify things and we say, here's our receptor. And it starts the pathway, and then we've got a series of neurons till we get up to the brain, and maybe we switch to another group of neurons as we leave the brain and finish on an effector. So all of this might be integration in the central nervous system. But at each one of those synapses, one synapse here, two synapses here, 3, 4, 5, 6, and then we have our final one in this example down at the effector. Say that's skeletal muscle, then we know that our neurotransmitter is going to be ACH. But the neurotransmitters in this pathway could be different. In the central nervous system, we use different ones. ACH is described as being excitatory to skeletal muscle. But the neurotransmitter, just for an example, the neurotransmitter in the central nervous system might be called glutamate. Don't worry about these names. I'm just showing you another example. All right, we haven't talked about this one yet. We're not going to talk too much about specific neurotransmitters, but neurotransmitters can be excitatory, like the ones we've been talking about all along, or they can be inhibitory. Now, excitatory is what we've talked about. Those steps that we drew on the other slide and what we talked about when we talked about the muscle excitation and contraction, those are all examples of when a neurotransmitter is excitatory. And if it's excitatory all the way along, then it means we're going to get a new action potential every step of the way. And in our example that we've got here, the result is going to be contraction in a muscle. Everybody follow me on that?
Yes.
So what would the role of an inhibitory neurotransmitter be?
They stop an action from happening.
Yeah, exactly. So when you think about integration, when you think about, if you go back and think about that example I gave you about driving a car and then somebody cuts you off. Well, some things you want to do, you want to stop, and some things you want to continue. So you want to excite and you want to inhibit. That's all part of integration. Some pathways you want to continue on, some pathways you want to block. So for example, think about that tack in the baby finger. Right? When the tack went into your baby finger, it caused pain. You're going to try to pull away from that pain. So if you think about it, you're going to pull. One way you might pull is you might flex at the elbow. Everybody with me so far? Because if you flex at the elbow, you're going to pull away from the tack. Everybody okay with that?
Yeah.
So the biceps is responsible for flexion at the elbow, right?
Yes.
And triceps is responsible for extension at the elbow, the opposite. So we want to inhibit the triceps and excite the biceps during our removal from the tack. So we would have somewhere along the line, we're going to have some inhibitory neurotransmitters signal the triceps to not contract. Does that make sense?
Could be an
Yes.
inhibitory.
So,
Go
sorry,
ahead.
can we say excitation and inhibition are happening alongside each other?
Well, they'll be in different pathways, but they will contribute to integration. Yes. So you're going to want to inhibit, in our example, you want to inhibit the pathway to the triceps while you're exciting the pathway to the biceps. I have a terrible time drawing a hand, but you'll get the idea here. Okay, so here's our terrible hand. Okay, we'll move our tack down into the heel of the hand here. So let's just draw in our tack right here. So it causes pain, right? And here's our humerus. We'll just draw the humerus in like this, and we'll draw in our forearm like this, just to be able to illustrate our muscles. Okay, so here's our biceps. And here's our triceps. Okay, does that work? That picture work okay for everybody?
Yes.
Okay, so our pathway, so this is where we start right here with our receptor for pain. And then we're going to travel in a nerve to the spinal cord. We're traveling into the spinal cord, and then we're going to travel through the brain and out again. And on the excitatory pathway, we're going to go to the biceps, and we're going to cause the biceps to shorten. When the biceps shortens, we're going to get withdrawal from the tack. Yay. Right? We're okay with that?
Yes.
Yes. Confidently, yes, or not so confident?
Confident.
Yes.
Okay.
Yes.
So now what happens though, what if we were excitatory at the same time to the triceps? If you contracted triceps and biceps at the same time, what happens to movement? They're opposite to one another. So what's going to happen to movement?
Sorry, I didn't get your question.
You mean flexion?
If we excite both the bicep and the tricep, what's going to happen to movement at the elbow?
Nothing.
Yes, nothing. And guess what that means? That means the tack is still stuck in our hand. So that's no good. So what we need to do is we need to branch off. And somewhere over here, we want an inhibitory neuron. These ones are inhibitory neurotransmitter. If these ones are all positive all the way along, right? Excitatory. Positive means excitatory. Plus, plus, plus. Negative means inhibitory. So we want okay, I got to cover this up. Whoops, that's not what I want to do. So what we want the triceps to do is relax, just do nothing, so that we can get our withdrawal. We can get our flexion away from the tack. So we got to go, this would be triceps that gets the inhibition. This would be biceps that gets the excitation, right? That's, now we're getting complicated. We're getting the integration where we have to send excitatory and inhibitory neurotransmitters in order to get the goal that we want, which in this case is to get away from that tack. So this is how we can change the signaling. This is how we can change the outcome to our response. We okay with that?
Yes.
Yeah. So when we look at neurotransmitters, let's just finish a little discussion on neurotransmitters then. So we're going to use the abbreviation NT for a neurotransmitter. So there's a few characteristics that have to be consistent with neurotransmitters. One, they have to be produced in the neuron. In other words, they've got to be inside the neuron to get started. And then once activated, so in our case, we said that the presence of calcium inside that axonal ending was activation, right? Once they've been activated, they get released. We're going to get a response because that postsynaptic cell is going to have receptors for that particular neurotransmitter. Okay, so when we talk about these neurotransmitters, there's some characteristics that have to be consistent in the nervous system. Produced in the neuron, and once they're activated, they can be released to produce a response on the postsynaptic cell, which means there must be receptors on the postsynaptic cell. We okay?
Yes.
Yes.
And then the final consideration is there needs to be a mechanism for removal, because sometimes that's part of integration, right? We stop signaling the next cell. So that removal might be that we have some enzymes that break it down. That's what degrading it means. Or we have a pump mechanism where we can take it back up into the neuron to be reused at the next signal. Reuptake, that's called.
Sorry, could you just say what you said again about the reuptake, please?
So there's two ways to get rid of a neurotransmitter in a synapse. We can break it down with enzymes, right, and destroy it, get rid of it. Or we can reuptake. In other words, the presynaptic neuron can take it back.
Thank you.
Okay. Okay. Now, let's just move away from these steps of excitation and all this sort of stuff. So all of this discussion has been on the special characteristics of the neurons, and the neurons are the players in the nervous system. But these neurons, as we already know, are very, very sensitive to their environment. We already know that they need a constant blood supply, or they're not going to be able to make up enough ATP in order to generate these action potentials and generate the production of neurotransmitters. All that stuff takes a lot of energy, right? So those are just some of the things. Now, in addition, they're really sensitive to pH differences. They're really sensitive to exposures to certain types of hormones or other types of proteins. So they need some protection. They need some other cell types that play a supporting role in the functions of these neurons. So these cells are called glial cells. And glial cells have a number of supporting. Now, you'll sometimes see them generally referred to, because of the nervous system, as neuroglia. These are support cells. So we have some support cells in the central nervous system. And what makes these cells different is, one, they don't have excitability, but two, they typically can undergo mitosis. So we can replace these with the same type of cell. So we're not as concerned about losing some to injury or that sort of thing. And considering that a big part of their role is protection, that's kind of a good thing. We can replenish some of these. So in the central nervous system, we have one type of cells called an ependymal cell. And ependymal cells, so that's ependymal cells are cells that basically are found in certain parts of the brain, and they're responsible for producing, they line the cavities that produce cerebral spinal fluid. So these are kind of like an epithelial tissue. Epithelial.
I'm sorry, can you repeat?
They line cavities in the central nervous system. So the center part of our brain have ventricles, spaces that are fluid filled, and those fluid filled components are lined with these ependymal cells.
So, can you repeat what you said responsible for producing which gland?
Epithelial cells that line the cavities of the central nervous system. That's what an ependymal cell does. And microglial cells, you can tell one thing out of that name. Are they big or small?
Small.
That's right. But they have a big job. These guys are part of the immune system. So they're big eaters, macrophages that are mobile, and they clean up the environment around the neurons. So either dead cells or chemicals that might be accumulating, that sort of thing. They're continually moving around, picking up cellular debris and waste and eating it, getting rid of it. And astrocytes are another type of glial cell. And they're responsible for providing what's called a blood-brain barrier. BBB. And the blood-brain barrier is a filter that protects exposure of the neurons to, for example, contents of the blood. Believe it or not, if neurons are exposed to blood directly, that will initiate cell death. They will die. That's partly what happens in a stroke, is a blood vessel bursts in the brain and blood is exposed to neurons, and neurons die from that exposure because a lot of the content of blood is toxic to a neuron. So this blood-brain barrier is created by these astrocytes, and they create a filter. They let some of the things from blood to the neurons, like oxygen and glucose, and some vitamin C and some other things that are good for neurons, but prevent other things from getting in, some hormones and some other proteins that would be potentially harmful to a neuron. So they provide a filter.
So is this filter essentially selectively permeable?
Yeah, that's a way to think of it, but it's made from cells as opposed to components of cells, like your membrane.
Okay, thank you.
So it allows nutrients in to neurons, but it prevents toxins. And then we got a mouthful, oligodendrocytes. Oligodendrocytes. These are neurons that help, or at least these are nerve support cells, glial cells, that support the neurons, give the neurons some structure and support, provide structure and support to neurons. They form something called myelin. Around dendrites and axons and insulate them. They provide this like a fatty wrap around those processes. And wherever we have myelin on a nerve process, the transmission of speed is faster and the neuron itself is insulated. So it's got some added structure and added protection. So that'll look a little bit like this. No, where'd it go? There. So let's get out of wherever I was. There we go. So what we're looking at here, if we were in the central nervous system in this picture, just hang on.
Question.
Hang on. This cell would be our oligodendrocyte, and it would wrap this fatty insulation around segments of our dendrite here. So providing support and structure, and the electrical pathway is much faster in a myelinated fiber than in an unmyelinated fiber. So we increase the speed of transmission and we add protection. Okay, question.
When are we going to go for a break?
Well, I definitely don't want to answer that question when we're in the middle of a topic.
Okay.
So we're close. So all I want to do before we go on our break is talk about the support cells that are found in the peripheral nervous system. So neuroglia, in the central nervous system, we talked about ependymal cells, microglia, astroglia, or astrocytes, and oligodendrocytes. And in the peripheral nervous system, the good news in the peripheral nervous system is that we're only going to talk about two in the peripheral nervous system that have very similar roles. So we won't have to repeat a lot of what we've just been talking about here. But we've got what are called Schwann cells. So those are similar to the oligodendrocytes. But in the peripheral nervous system. So they form the myelin sheaths and they increase transmission. Okay, the one difference is in the peripheral nervous system, the Schwann cells can encourage dendrite and axon repair. The oligodendrocytes in the central nervous system cannot do that. So that's a little exception to the rule as far as that goes. And then we have satellite cells, which are similar to the astrocytes. In other words, they form a bit of a barrier. It's in the peripheral nervous system, it's not going to be called the blood-brain barrier for obvious reasons. Peripheral nervous system doesn't include the brain, but it does provide a bit of a filter. These satellite cells provide a bit of a filter to protect the neurons in the peripheral nervous system from exposure to toxins and so on.